So here we want to look at the sort of scientific approach to an understanding of what it means to be human, and that means biological or maybe biophysical anthropology. So human origins, evolution, and I want to get eventually to the Human Genome Project itself, which is sort of the culmination of this effort of biological science anyway to understand what it means to be human, and then talk about comparative genomics, and then finish up with a little bit of what I promised about the brain evolution before we get on to the philosophical piece after lunch. Historically, anthropology has its origins in sort of the Enlightenment effort, if you will, to understand the natural world in all of its glory. And so even though the earliest Western speculations involved people like Aristotle and Thomas in what I'm going to call philosophical anthropology, the scientific enterprise doesn't really begin until much later during the Age of Exploration, when especially Europeans begin to encounter the other, okay, to go out into the world, especially into the Western Hemisphere, the New World, and find other beings, other humans, and that were different than them. So this led to a lot of speculation. For instance, the myth of the noble savage, Hobbes's Leviathan, for instance, writes about this, or in Rousseau's Discourse on the Origin of Inequality, he writes about these others and their role and their culture. And so speculations began then about these others, these people, and with relationship to an understanding of people in Europe, mainly, who were the ones writing these and doing the exploration at the time. Now, as I said, biology itself was pretty much an observational discipline throughout the time that physics and astronomy were maturing and blossoming into really mature sciences. I should say that the definition of a mature science is that it has a theory and a language. Okay? So physics had, of course, Newtonian theory, and the language is mathematics. Okay? Biology had no theory and no language. So it was not a mature science by any definition. But it was observational. People were observing the living world. And it fell to Carl Linné, the Swedish physician, to finally come up with a system of classification. As people sometimes say, God created nature and Linnaeus gave it order. Back to your point about looking at what is chaos and making order out of it, right? So Linnaeus comes up with this system of classification in which humans are classified as primates. And actually, Linnaeus is his Latin name because he wrote in Latin, but Carl Linné had a lot of trepidation about classing us as primates, putting us with the monkeys, okay, and the apes. And he had some theological issues around that he worried about. But nonetheless, he said that when I look at this descriptively, and order things in terms of how they look to me, their detailed, descriptive, biological structure and function, humans belong in the Homo sapiens, but part of the family Hominidae, but in the order primate. Now, he makes us genus Homo, species, of course, sapiens, Homo sapiens. We are in the kingdom Animalia, phylum Chordata, subphylum Vertebrata, class Mammalia. And just as kind of an aside, why did he choose the class name Mammalia? Why is this class ordered and called Mammalia? Well, they're animals that nurse their young. But why name the order after that? Interesting speculation, and some historians of science have asked about that because they said, well, gee, only half the members, only the females, have mammary glands, right? So why name the whole class Mammalia? And it turns out Linné, as a physician in Sweden, was a proponent of breastfeeding for the health of infants. And he was part of a movement in Europe that was countering the women who didn't breastfeed. And he was convinced that healthy infants, as we all know now, healthy infants would be better, or infants would be healthier if they were breastfed. So to drive home that point, he named the class Mammalia. And this is in his writings, just as an aside, in case you were wondering about why we're mammals. Now, this classification scheme finally gave biology some kind of theoretical foundation. Okay? So biologists now, botanists and zoologists, begin to look at the natural world with respect to this ordered scheme. And this began to get people to think about how this came about. So in terms of our particular component of this, it gave birth to what is called physical anthropology. That is, making physical measurements of organisms, in this case humans, right? Anthropology. People were measuring all kinds of organisms because that's how you fit things into the scheme. When you discovered a new organism, you measured it. You got its characteristics, its dimensions, other things about it, and you were able to fit it into the Linnaean scheme. So craniometry came into being. People started making measurements of human components to try to see how they fit into various schemes. It provided a theoretical framework, in fact, for Darwin. When Darwin went out from England on the Beagle, on this voyage, he had already, of course, you remember, he was going to train for the ministry. Okay, actually, before that, he was going to be a physician. He went to Edinburgh. His family trade was medicine. So he was going to go become a physician, but he couldn't stand the smells of the anatomy lab. So back to Cambridge he came and was going to train for the ministry until he got under the influence of the biology tutor and really fell in love with the living world as a biologist. Was convinced that he could take this post on the Beagle as the naturalist and took off. Now he took with him on that voyage a lot of scientific books. Lyell's Geology, for instance. But he took also books about cranial measurements, about measuring the characteristics of organisms, because that's what he was going to do. And of course he came back with all of these data, fossil record, measurements of organisms that he encountered, both humans and others. And out of that grew this great theory that literally was another turning point in science, especially in biological science, because with Darwin's publication of Origin of Species, we finally had a theoretical framework that biology could take. In other words, with Darwin, it suddenly was not that everything you see in the living world was created as we see it, but rather everything evolved from a common ancestor. And that becomes one of the central two pillars of modern biology, along with Mendelian genetics. And we'll come back to a little bit about that piece in a minute, and say more about the influence of Darwin on this whole process. So with Darwin, though, comes the search in the fossil record. Let's see what's been preserved when we dig up areas where these fossils can actually exist, see what's been preserved, and try to track through where it goes in a schematic way, again using the Linnaean classification system, but now with an evolutionary view that we're looking for forms that pre-existed us, so-called ancestral forms, if you will, from which organisms may have evolved. And this leads to this search for the evidence of our own history, the primate fossil record. And if we're still looking for this, as you know, we're still discovering the fossils, the so-called hobbit fossils in Indonesia, these very small people that were recently discovered that are part of our primate lineage. And all of this now begins to fit into a pattern of what's called physical anthropology, the search for human origins through the fossil record. Okay? So we see that when we sort of start to order these in some structural ways, they fit the model of descent with modification, which is part of Darwin's model, right? So we have a model now to work with, descent with modification, and we now start looking in the fossil record. And we're finding evidence that kind of fits with that model and supports it as a scientifically valid way to look at Homo sapiens. Now, as that's going on, and that, of course, begins to take place around the time of Darwin and really becomes very robust at the beginning of the 20th century. But the Darwinian model itself isn't that successful scientifically at first, because Darwin really didn't have an understanding of genetics. He didn't know how inheritance worked. The predominant idea during Darwin's day among naturalists was that inheritance worked by blending. So the characteristics of the two parents are blended in the offspring. A classic description of that is if you cross a white-colored snapdragon with a red-colored snapdragon, all the offspring are pink. So that was considered blending. Even though we know now the Darwinian explanation, or the Mendelian explanation of that, it appeared to be blending. Now Mendel was sitting in Austria proving that inheritance was quantitative, particulate. There were laws that he published in 1868 in his famous paper on inheritance, but nobody read that paper. Mendel actually read Darwin's book. We have, in fact, in the Mendel Library, in the Mendel Museum in Austria, in Brünn, his copy of Origin of Species with his notes in his own hand. Okay? But nobody read Mendel's paper. Darwin mentions Mendel in one of his later works only as a botanist, nothing to do with genetics. So Darwin's theory of descent with modification and selection fell on deaf ears scientifically, never mind the theological issues, right? There was an engineer in London named Fleming Jenkin who wrote a paper critiquing biological evolution in Darwin's model, saying it couldn't possibly work because any selective advantage that a trait would have would be blended out over generations. So how could you select for it? How could it be selective, right? And he published this paper, and in fact, in the 5th edition of Origin of Species, Darwin spent about 6 pages defending himself against Jenkins' arguments. And he had no way of doing it other than to fall back on the arguments of Lamarck about acquired adaptation. It's the only thing he could do. He spent the entire book attacking Lamarck, except he comes back to use Lamarck to defend himself against Jenkins. And it's because he didn't know genetics. So it was falling on deaf ears until the beginning of the 20th century, when a number of scientists rediscovered Mendel. They were doing experiments with flies, with a variety of organisms, discovered these rules of inheritance, looked back in the literature, and sure enough, 50 years earlier was this paper by Mendel. That's why we call it Mendelian inheritance, not de Vries inheritance, right? And they acknowledged that Mendel was the origin of it. So by the middle of the 20th century, you put the two things together, especially on a population level, and we see that now biology has a theory, descent with modification from a common ancestor, and a way of explaining inheritance in a quantitative fashion, the Mendelian laws of genetics. We actually call them laws, same way we call them laws in physics. And ultimately, by the middle of the 20th century, we even have a language for biology, A's, G's, C's, and T's, okay, the sequence of DNA. So we become a relatively mature science by the middle of the 20th century. And it's at that time that the approach to looking at humans takes on its molecular direction. Okay? So we have the rise of what we call molecular anthropology. The use of these biological tools, especially genomics and genetics, to examine human beings, right? We're asking the question about humans. Here's Jonathan Marks, very interesting man at University of North Carolina in Charlotte. He wrote a book, What It Means to Be 98% Chimpanzee, Apes, People, and Their Genes. A very nice book that sort of is about the rise of molecular anthropology, the use of genomics and genetics to analyze the human genome and its relationship to other creatures. So I recommend it as a book. It's a little old. It was published in 2002, but still worth a read. Now I'm going to step through some examples of how molecular anthropology has worked to talk about the human population. For instance, we learned in the early part of the 20th century that our blood has a variety of markers that can be analyzed very easily. These are proteins on the surface of cells in our circulation, especially the red blood cells. One of the most common is called the ABO blood group. I'm sure we all know our blood group, right? And it's inherited in a strictly Mendelian fashion by virtue of a number of enzymes which either do or don't modify proteins on the surface of the red blood cell to produce cells that are antigenically A, B, or O. O means that they don't have the modification on the surface, the null allele. So we have 4 blood types. And people have used this in an anthropological sense to define populations. Okay? So you can find studies where people have gone around the world and looked at the distribution of these genes in the human population. For instance, and the colors don't show up very well, the B allele accounts for about 16% of the human population, and you can see that it's mostly over in Asia, with some representation in Africa, very rare in the New World, the B allele. The A allele, on the other hand, is a little more broadly distributed in the New World, especially in the northern part of North America, but again, heavily represented pretty much in Asia, and it's 21% of the human population. The O allele is much a New World allele. It's very heavily represented in the New World. The null allele is 63%. And so that the combination of those are what give us the A, B, and O blood types. And I won't go into the genetics, but this is the kind of study that's done at a molecular level now to characterize human populations, right? To start to say something about who we are and how we maybe differ genetically. One major question that came up early on is, are we all the same? When the explorers were going out to the New World and encountering indigenous populations, are these people human like us? We're from Europe, they don't look like us. Gosh, are they really us? So, in fact, there were arguments. I don't know if you know the name Bartolomé de las Casas, a famous Dominican, who went back to Spain and defended the idea that the indigenous people of the New World were actually human beings, because the argument was being held at Salamanca, maybe they're not human. Maybe we don't have to treat them as human, okay? And de las Casas wrote a huge tome on the indigenous peoples of Mexico that he had encountered and defended that position back at Salamanca. So, but that question had persisted. Where did the human population come from? So now we can apply molecular tools to that question. We can examine the human population for commonalities of genetics, using, again, our model, descent with modification and genetics, right? So one of the great ways of doing this is to examine some components of us that speak to our inheritance. One of them is the mitochondria. These are the energy parts of the cell, the little organelles that originally in our evolutionary history were a separate organism, another bacteria that was engulfed by another cell and then became a captive endosymbiont to make energy. That's how we make our ATP. But they have their own DNA, which speaks to their origin as a separate cell in that endosymbiotic model. They have their own DNA. And for humans and other mammals, we inherit our mitochondria in a very interesting way. We get all of our mitochondria from our mother. The mitochondria that you have were originally present in your mother's egg, and the sperm, which provided the genetic component from your father, that sperm had its mitochondria left out in the fusion. So by and large, when the sperm fused with the egg cell, all of the mitochondria that are left were maternal. Okay? So we all share maternal mitochondria in that sense. So what some scientists did was say, well, then let's look at the DNA of human mitochondria from people all around the world. And it's a project that was started by a group in Stanford. So they literally went around and collected mitochondrial DNA. It's relatively easy, a little skin sample, maybe even a hair root. just to get some mitochondria. If you've got the tools, you can do this, right? And then they said, let's kind of see how we're all related then. Let's make a tree out of this, a phylogenetic tree. We'll kind of see how everything relates. And when they did that, this is an example of one, they all root to a common ancestor. So the idea became that, yes, all of us, in all existing populations on the planet, and this database has now been extended to be huge, descended from one or only a few females. So she is metaphorically called the mitochondrial Eve. Okay? Not because this is proof that Genesis was correct, that's not the issue here, but this is indicating that all currently existing human populations came from one group of ancestors who probably went through what we call a bottleneck, a genetic bottleneck, where the population narrowed to a small number and there were only a few women present, and all living humans descended from that population. Okay? And this is now commonly accepted as a model. Now, there were a lot of arguments about it when it first happened, not because the data didn't indicate this descent, but rather the timing issue, which has to do with the rate of mitochondrial change that was being observed here. And the timing issues of this population of our ancestors was at odds with some of the more traditional anthropological dating information, like radiometric dating or fossil dating and layers and the like. So there were a lot of arguments in the anthropological community about the timing here, but the principle was not at argument. We all descended from this common ancestor. And there's another way of looking at our ancestry, and that's through our Y chromosome. So as you probably know, women have 2 X chromosomes, but all human males have an X and a Y. And the Y chromosome is the male determining chromosome. Our body plan is female, upon which maleness is imposed. I always love to tell my students that, because the girls all go, yeah, and the guys go, huh? So, but it is true. Without the Y chromosome, the human body plan is female. The Y chromosome imposes maleness on it. So we inherit the Y chromosome exclusively from our fathers. So all of us have our Y chromosome we can trace back through our fathers. And any problems in my family are blamed on the Y chromosome. For me, my son, my grandsons, it's all my fault and my father's fault. Okay. So people did the same thing. Look at the Y chromosome from people all around the world, men all around the world, and relate them, and you get exactly the same answer, that everybody roots to a common set of male ancestors, one or a few male ancestors, and predictably this was, even though it came later, so it's now called Y chromosome Adam, that person we descended from. So with the same result. We came from this bottlenecked population. Now, yes, people who want to find scientific evidence that Genesis is true leap on this and go with it. You know, I'm not going there with this. This is simply the data. Okay? Maybe I could argue Genesis reflects an ancestral story about this time when we came through that Garden of Eden. Okay? Maybe that's what we're looking at in Genesis, is a sort of ancestral memory of that, because there are origin stories all around that have similar kinds of flavors. But nonetheless, biologically, we come like that. And so now we think that the whole world is populated with humans who came from these original sets. And so there are models that are being drawn, because it turns out those original ancestral humans, either the Y chromosome Adam or the mitochondrial Eve, existed in Africa. That's where they were. And I have to tell you a funny story about this. I was in Tucson at the university, and a secretary of my department asked me if I'd come one morning and give a talk for, I don't know if it was some like Rotarian club or some group of businessmen that met for breakfast in the morning, right? And so I came and I decided to talk about, at the time it was mitochondrial Eve, the mitochondrial thing. So they're all sitting there, and you know, I have 20 minutes after breakfast to say a little something about science at the U of A, and I tell them about this story. And they're all just kind of, okay, is it time to go yet? scientist is talking. Okay, I can sleep some more. And I got to the end of the story, and I said, and so our ancestors, that ancestral mitochondrial Eve, came out of Africa. And this guy in the front row sits up and folds his arms and looks at me, and he says, great accent, you mean to tell me we all came out of Africa? And I said, yes, that's what I'm saying. He sat back and he said, huh, and just staring at me. So I'm going, oh, look at the time. I really have to go. I think I better get. He was just outraged. That just did not start his day off well. But I'm sorry, that's where the ancestors came from, Africa. So there are various models for how the populations from Africa spread out to populate Europe and Asia, and of course we have the model that our indigenous population here came ultimately over the land bridge, right? The Alaskan land bridge from Asia. Now, these kinds of data like this are embedded in our scientific models, but they get used in some very interesting ways socially, right? So there's been a kind of an indigenous reaction to this. Many of you may have known Vine Deloria, who passed away recently, as a phenomenal author and thinker in the Indian community. He wrote a book called Red Earth, White Lies, and another one called Evolution, Creationism, and Other Modern Myths. Vine was a theologian by training, but was also a lawyer. And Vine's point was that these origin stories that we tell, which have these anthropological data embedded in them, get used in very interesting ways against indigenous people. So there's a famous case of the Indians around Pyramid Lake in Nevada who were claiming ancestral rights to certain lands that were going to be exploited by the government. And the argument was made by the lawyers in opposition to their rights that said, your people only came across the land bridge a few thousand years before we got here, so you don't have any more rights to this land than we do. When the Indian origin stories had nothing to do with the land bridge. Okay, so there's that. Another good one is that the Clovis site down in southern New Mexico is the one that anthropologists use to make the argument that the indigenous people killed off all the megafauna in North America. Because there's a Clovis site where they were hunting mammoths, right? And so they make the argument that the disappearance of megafauna in North America was the result of overhunting by the indigenous people. Now, go over to Europe. Megafauna disappeared in Europe at the same time. Why? Disease, not overhunting. So Vine makes the point in this book that there's a kind of ethnocentrism going on with these stories. So just to let you know that not everybody is ready to jump on the anthropology story here. All right, so that's what was going on, these molecular approaches to understanding who we are as people. Now along comes the Human Genome Project itself. And the project originally was a project of the Department of Energy. Okay? They had the money to do this. And they called a meeting in Alta, Utah, brought together all of the molecular biologists, this was in early 1980s, and said, technologically now, you're sequencing DNA at a good pace. Would you, if we gave you literally all the money in the world, be able to sequence the human genome? And the molecular biologist president thought about it for a nanosecond and said, yes, we could do that. And this was the first big science project of biology. Physics had done its big science. We'd never done that before. So this was going to be big science. Now, why the Department of Energy? Okay. Well, it all comes back to here, right? It turns out that when we developed the bomb, Fat Man and Little Boy, and dropped them to end World War II on Japan, on Hiroshima and Nagasaki, the US government was concerned that in the coming Cold War that they foresaw, we were going to lose Japan to the Russians. So in order to keep the now subjugated Japanese nation in the Western fold, they consciously started a project to examine the effects of radiation on populations using studies of the people in Nagasaki and Hiroshima. And they called this the Radiation Effects Research Commission. Okay? And it was sponsored by the Manhattan Project. It was a subunit of the Manhattan Project. There are great pictures online of Eleanor Roosevelt visiting in Hiroshima at the laboratory that's doing the data gathering of health effects of radiation. And the idea was to figure out what are the effects of radiation on human genetics. And it turned out if you look grossly, you couldn't really see that. So the Radiation Effects Research Commission was all interested in finer and finer levels of genetic analysis. Of course, the Manhattan Project became the Atomic Energy Commission, became the Nuclear Regulatory Agency, became part of the Department of Energy. And all along, the Radiation Effects Research Commission, it was actually originally called the Atomic Bomb Casualty Commission, was part of that, the ABCC. Okay, it still exists, now called the Radiation Effects Foundation or something like that. They're the ones that sponsored the DOE project. That's why the money was DOE. It soon became NIH, though, because the National Institutes of Health said, wait a minute, we do human health, that's what we should have. So they basically landed the project at NIH, and the goal was to get a complete sequence of the human genome. And it was completed in February 2001 and published in Science and Nature, put online. The whole database thing became the key. Now, there are problems that arose immediately, okay? The people who started the analysis were people like Jim Watson, the discoverer of the DNA structure. And his stated issue was as follows. Once we sequence human DNA, we'll know what it means to be human. Jim is the ultimate materialist, right? The ultimate reductionist. Once we sequence the DNA, we'll know everything about what you are. No questions asked, okay? Jim assumed that there'd be something like 100,000, 90 to 100,000 genes in the human genome that make us human, right? First problem. When we sequenced it, we only have about 25,000 genes. Even worse, we don't have any more than the fruit fly or the worm, okay? Any metazoan. We have about the same number. Suddenly, the model is in jeopardy, okay? Philosophically in jeopardy. The other issue that came up when the database was populated with these data is that the data are so immense in size that defining what a gene is becomes essentially impossible. And so the concept of gene, which was at the heart of doing this, became a challenge. What does it mean to say that this is a gene or not a gene in the sequence? Which is now an epistemic challenge. So it was an ontological challenge and an epistemic challenge, both of which pretty much brought molecular biology up against a wall. And I'll describe, when we talk philosophy afterwards, what happened that rescued us, but also changed us philosophically, okay, in a dramatic way. So we're in the middle of kind of a paradigm shift now in biology. We'll come back to that philosophically later. But we now have a powerful tool for studying humans. It's called genomics. Okay? So we can not just sequence, and the sequence, by the way, was not one person's. It was a compendium of sequences depending on who had DNA from which chromosome in which lab around the world. So in the end, the sequence is maybe, I think at most, a dozen people. Okay? But we now have a tool where we can do anybody's DNA. Okay, we know how to do this now. And we can do it very quickly, and we can do it in sort of interesting ways. So for instance, we can do what's called haplotype mapping, okay, where you don't do the whole sequence, you actually look for differences along a chromosome in specific locations, and then you look how different populations of humans do or do not have certain changes along the location of the genome. And they're called haplotypes because it's either the maternal or paternal component of the person, the haploid component. Okay, so you can do this very quickly using some really neat enzymatic techniques. and start to accumulate variations that are due to single locations, so-called single nucleotide polymorphisms, or abbreviated SNPs, often called SNPs. Okay, so we can begin to make maps of these haplotype SNPs and look at what happens in populations. And we can use these maps in some interesting ways. So here are some data from a study on high blood pressure in Texas, showing haplotype frequencies divided up by African American, Japanese, Latino, Native Hawaiians, and whites. Those are the data shown here. And the sequences you're seeing are just regions of DNA that have these differences, okay? So what's being used here? The positive outcome of these kinds of studies is called customized medicine. So for instance, different people have different reactions to, say, high blood pressure medication, right? So if we know your haplotype, we can customize drug delivery. You may need more for your haplotype compared to someone else to effectively lower your blood pressure, or you may need less. So we can actually do customized drug delivery if we know your haplotype. That was the goal of this giant project at NIH called the Human Haplotype Map, the HapMap project. So disease prevalence and genotype are the goal here. Now, the problem is it reintroduces something called racial characterizations, or racial designations, that we thought we had lost. Anthropologists had given these up as nonsense descriptors back in the 70s. In the 70s, had written that saying races doesn't mean anything anymore. That's why we went to ethnicity, right? Because it doesn't mean anything genetically. But here we are reintroducing racial characterizations. In these data I'm showing you, people who were recruited into the study at Texas were asked to fill out forms self-describing their ethnicity. But then when the data were published, they were published as racial types, to reintroduce the word. So there are people who are very worried about the use of these data and the HapMap itself. Just worried not in the medical outcome, but in the sort of social, cultural sense of what's going on. But they're powerful tools now, so we can do what's called comparative genomics. We're sequencing organisms at a huge pace. Laboratories around the world, I think almost monthly I see published in Science or Nature another edition of a complete genome to a database. So this one's out of date already. This is just some of the ones that have been put into the database, the complete genome sequences. And let me just highlight the human, the mouse, and the chimpanzee, which are in the database, because we can do some interesting comparative genomics here. That will give us some great potential. For instance, there's a mouse model of Duchenne muscular dystrophy that's becoming very advantageous for studying the human form of this disease. Because it turns out the protein that is mutated in muscular dystrophy, Duchenne, in the mouse is exactly the same as the one in the human. So the consequences of that mutation in the mouse can be examined in terms of drug interventions and all sorts of things. But even more interesting is the chimpanzee. This is our closest relative on the planet. 2% difference in genome, okay? And so a lot of things about humans and chimpanzees could be useful. So for instance, in this table, we see that there are a variety of human conditions that are very different in our nearest relative. For instance, HIV progression to AIDS is very common in humans, but very rare in the great apes. Okay? Hepatitis complications, very severe in humans, mild in the apes. Let's go down to myocardial infarction, which is common in humans and rare in apes. Okay? And the list goes on. So people are looking at things like this to say, gee, what can we learn by studying the great apes genome and the subsequent effects on their biology compared to ours, because we're so close. Okay? Now, we're close, but we're not identical. And this is a key aspect of who we are as humans, genetically. Humans and chimpanzees are close. And there are people who want to make chimpanzees primitive, simple humans, give them rights, Okay, there's a movement in Europe called the Great Ape Project, which is actually seeking to legislate status for chimpanzees that are equivalent to human rights, right? And so, is this real? Our genomes are similar but different. For instance, if you just look at the chromosomes, we have this set, our 22 plus our X and Y and our mitochondrial genome. So we have 23 pairs. Apes have an extra. This chromosome we have 2 is divided in 2. So somehow, evolutionarily, they went down a different path. And to say that we're 98% the same ignores the reality of the fact that we're 2% different. And this is interesting in the New Scientist in 1999 that says a creature that shares 98.4% of its DNA with humans is not 98.4% human any more than a fish that shares 40% is 40% human. It's a sequence comparison. So we have to watch this question of misplaced reality of the result here, because chimpanzees are not humans in any sense of the word, even though we share enough that our genomic comparison could yield some very important medical advances for us. We understand, for instance, how Alzheimer's occurs in us but not in the great apes. We don't see the neurofibrillary tangles in great ape brains. All right, so what makes us different? If there's only 2% genomic difference, how are we really different? And we're going to argue in the philosophical section after lunch that we are qualitatively different. Thomas would argue we are qualitatively different. Here, the scientist wants to argue there's a gradation and maybe not that much qualitative difference. However, Terry Deacon, who I mentioned earlier with his book, The Symbolic Species, wants to argue that we are qualitatively different, and what makes us different is our evolution of language and the use of symbols, okay? That makes us different from even primates that have language, okay? Have some sort of language. And he's been struggling since he published this book to explain himself, because he is very much at odds with the people who have taught apes, parrots, other animals to actually communicate. So we had a woman in Arizona who had a very famous parrot who lived for many, many years with her, and an African gray parrot who she could communicate with. She had taught enough symbol language to that Alex could actually talk to her in English. Okay. In fact, the day he died, he supposedly told her that he wasn't feeling well and was probably going to die. Irene Pepperberg is her name. So people have done a lot of this work especially with primates, especially the great apes. But the issue that Terry says is even in the best case, the apes don't really acquire language in the same way we do. That is, they don't have brains that can be self-reflective. They don't have brains that can do abstraction, the intellectual activities that we're famous for. So he wants to know what makes us different, even though we share this evolutionary history. So he then argues that the model has to be language evolved as a process of evolutionary change, and the human brain was subject to selective pressures like every other part of us. However, and this is the part I was alluding to at the end of the last one, Deacon argues that when we changed in behavior to become tool users, and especially when we developed into hunter-gatherer communities, that relieved a certain amount of selective pressure that we were under for survival. Okay? It also added the necessity for communication in a community to be a successful hunter-gatherer community. So he says that the complexity of the brain for language evolved due to the release of other selective pressures. And there's evidence for this in birds. There are certain birds that in captivity, in the wild, where they're foraging as all other birds are foraging, have a very limited song set. But if you take the same bird ancestrally and put it now in captivity and give it food So you release it from foraging, release it from the pressure of having to survive, its song repertoire greatly increases. And you can actually observe this anatomically in the complexity of the bird brain as it increases over the generations of that population. And these species have diverged now, obviously. So Terry argues that our brains have done this, that our brains have become more complex because becoming successful tool users and entering into community released us from the pressures of survival as individuals and gave us the opportunity and forced us in the direction of complex use of symbols, especially in communicating about hunting. All right, so that's his sort of key that he wants to use as the thing that drives us towards this symbol-using, language-evolving creature, right? Now, that's just his biological explanation, and he's defending it. And he has a lot of critics. So whoa, that's going to look good, Casey.
I'll put the slide.
Okay, thank you. The camera kind of went sideways. That's his explanation that sort of gets him into the biological mode of discussing this, right? So he can say, all right, I'm with you evolutionarily, even though I don't agree that teaching the chimpanzee to recognize what you've got it recognizing is really making it like us. We're different. And we're different because we've been through this process of making community, of becoming tool makers and tool users. All right, so where does that now bring us in terms of the human person? We've got a genome sequenced. Unlike what Jim thought, we're not very different from other metazoans in terms of numbers. Wow, okay, that's interesting. We can do comparative genomics, so we know how we're different and how we're the same as other animals, and that's leading us into some great medical directions. We know we use language, and we know we use it in very different ways than other animals. And by the way, I don't want to be anthropocentric about this, and I don't want to also denigrate the work that's been done with the other animals. I will say that no one really knows if dolphins talk to each other and understand each other the same way we're talking now, because you can't be a dolphin, okay? All we can do is intuit from their behavior, okay? And we know they're highly intelligent creatures, and we know they can do certain things in a very great way that we can't do even. But all we can do to speak about consciousness is look at each other, okay, and ourselves. So yes, it may be that if I understood the mind of a chimpanzee, I would be really impressed with their ability to do abstract thought, even though they can't speak to me about Einstein's equations, right? I just don't know that piece. So Terry is admitting that, but saying, given my study of humans and language development, I have to say we're very different. At least we behave differently in terms of how we do this. And that's led us to sort of this what David Chalmers, the philosopher of science, calls the hard problem. Understanding human consciousness. What do we mean by that? Okay. In fact, we now talk along David Premack's lines, another behavioral biologist, about theory of mind. How do we conceive of our mental states? And, more importantly, how do we infer mental states in others? Okay? The key component of this is really at the heart of what it means to be human. Okay? Because we can say all we want to about our genome and our comparative genomics and about our craniometry and everything else we've talked about. Biologically, the key component of what it means to be human is what you're doing now. Okay? Which is listening to me communicate to you some sets of ideas, and you're thinking about them, and you're inferring mental states in me, and I'm inferring mental states in you, and we have an I-thou relationship going on that is at the heart of what it means to be human. And so the theory of mind that we all have, we share right now in this room, is such that it allows this to happen, right? And so the issue really becomes more philosophical at this point than it does scientific, although you're going to see in the philosophical piece that we do after lunch how the science of human consciousness has led us back to the atomistic kind of level, where people who are studying consciousness at the level of neuroscience are doing it by imaging the brain. Clearly, the brain is involved in our consciousness. We know that in a number of ways, both our own personal knowledge as well as scientific knowledge. In terms of brain damage, in terms of anesthesia, in terms of lots of things, we know about consciousness and the importance of this organ system, the central nervous system, in that phenomenon, right? So obviously, if you're a scientist and you have a tool that can image the brain, then go image it and find out what it's doing when you're doing certain tasks, right? So functional MRIs, we'll talk about later, as a key aspect of that influences the philosophical piece. But at the heart of this is really this issue of how I conceive of my mental states and how I infer your mental states in the process of our interrelationship. There's a lot of what it means to be human. So let's stop there, see if there are any questions about this scientific piece. And so the question is, are we truly free from evolutionary pressure if we move from individual life to community life? Is that a fair way of summarizing it? And I'm going to say you're correct in that but it's the adaptive, selective advantage of being able to use symbols that then drives the complexification of the brain, Terry's saying. Terry is arguing that the community formation, the use of tools in community formation, relieves the foraging, hunting pressure. So survival is no longer dependent on my ability to run down that animal and kill it by myself and eat it. Rather, there's a group of us and we have tools. So we're being better fed now as a community using a spear compared to me running down with my bare hands, right? So that selective pressure is gone, and now there's an advantage to vocalization, to symbol use, to having a theory of mind. That whole piece, he says, now is free to evolve in a way that the brain becomes more complex. I think that was his comparison to the bird, was that as the bird got free from having to flit about in the forest and find available food, is now in captivity where food is readily available, the song selection became that much more complex, and the brain developed a complexity as a kind of a, not direct comparison, but as a kind of an analogy to the human condition.
So you're saying then that really what's going on is a change in the selective pressures, right?
Yes, yes, yes. Not a complete release from the pressure to hunt and be successful by yourself, with no tool or implement to help you, or community. Yes.
Are there any studies trying to find consciousness, so consciousness in animals? I guess that is.
We have to define what consciousness means first, which I'll try to do after lunch in the process of getting to the philosophical piece. Because what that word means then dictates how you go about looking for it, right? So if consciousness in its broadest sense means sentience, okay, awareness of environment, let's say, then clearly animals have that. My dog Molly knows where she is, okay, and knows me, and she's aware of a lot. She's conscious. When she's asleep, she's different than when she's awake. Compared to our kind of consciousness, which is self-reflective, abstract, which is a different level. So measuring it in animals is easy if you define it in a certain way.